That brings us to our the last speaker of the session, which is Alan Andersen from Charles Darwin University, who's going to talk about fauna restoration standards at Ranger Uranium Mine, I think.
Thanks, Dave. Good afternoon, everyone. The presentation I'm about to give is based on a project that was part of the Northern Australian NESP hub in collaboration with the Supervising Scientist branch of the federal government and ERA, who are the owners of Ranger Uranium Mine. One of the key principles of best practice restoration is the identification of clear standards and specific indicators that tell you whether or not your restoration has been successful. And that's what my presentation is fundamentally about. It's really important to do this for two key reasons. The first is to provide industry with clear goalposts so they know exactly what they have to achieve. And the second is it provides very clear criteria for government, the regulators, to be able to assess whether or not restoration has been successfully achieved. And then to relieve industry of its ongoing obligations. So we've heard a fair bit about this, haven't we? That the history of restoration ecology really is all about revegetation, and fauna gets little mention, except for this particular symposium, which is good. And we've also heard about reasons why one should include fauna, and it can range from, well, they've got intrinsic biodiversity values. We've heard a lot about functional roles in ecosystems, and particularly how they promote ecosystem restoration. But we obviously need to be mindful of that they're important in their own rights. And we've also heard the idea that revegetation, successful revegetation, does not necessarily equate to successful faunation, the old field of dreams we've heard about. And a couple of reasons why. One is that just because a habitat might look fine to us, if we measure all the vegetation variables, it doesn't mean it's fine for the animals. But also, there is significant dispersal limitation issues that we need to be mindful of. And so, it's what a young wise person once said back in 2012, the development of faunal standards is the next challenge in restoration ecology. And one of the major challenges for identifying faunal standards is this one, and that is the issue of detectability. Because we all know plants, they're there, go and measure them, they're not running away, hiding, being difficult to find. And that most animal species are relatively uncommon, and very, very many of them are difficult to detect. And often, more often than not, they have to be captured rather than directly observed. And most techniques for capturing animals have their own intrinsic biases. And so this issue of detectability is really fundamental to the issue of identifying faunal standards. I'll be repeatedly coming back to. And so what I want to do today, two things. First of all is present a general framework for developing faunal standards for successful restoration. But then secondly, use Ranger Uranium Mine as a case study, where I'll be drawing on some information that we've collected that's specific for Ranger in terms of recommending standards specifically for Ranger. We've heard a lot about Ranger Uranium Mine, whole sessions of it yesterday, but just very briefly, the key issues is that it is surrounded by World Heritage Kakadu National Park, World Heritage because of both cultural and natural values, and so the standards expected of the restoration are exceedingly high, and the expectation, and in fact the agreed goal, is the site, the study region, needs to be restored so that it is a similar ecosystem to that in surrounding Kakadu National Park. And if it's to achieve that goal, the faunal communities also obviously need to be similar. So here's the framework that we've developed and we'll be working from. The first task is to identify the appropriate reference conditions from which you judge ultimate success. Second goal is to identify what species you include in those assessments. The third goal is to identify what aspects of those species that you measure to include as indicators. The fourth goal is to specify what is the level of similarity that is acceptable. What does similar mean? How similar? And the fifth one is to specify a robust sampling methodology so that then you can reliably sample the fauna and make reliable interpretations of those results. And I'll just go through each of them in turn. Appropriate reference conditions, and that can come at a number of levels. The first level clearly is whether the ultimate goal is more ecological restoration, where biodiversity values are a key consideration, or is it more rehabilitation, just restoring the functional roles in a production context? And they're obviously, the standards and indicators for each of those will be very, very different. And the next challenge is, let's assuming that it is ecological restoration. In most areas, there's a wide range of biomes occurring locally. So which one is the target one that needs to be identified? And there's the caveat there at the bottom, as in that will often be constrained by the final landform of your restored landscape, and that sometimes the final landform will not be able to support a particular biome in the area. So in terms of Ranger, the agreed reference condition is the regionally dominant savanna woodlands that surround not just the Ranger Uranium Mine, that dominate the broader region and in fact dominate all of monsoonal Australia. The second part, well, which species to include? And here is the problem of detectability. And the idea here is that there's no point in including in your species to assess those species that you just can't detect reliably and get reliable information on. And so there's a lot of effort now looking at occupancy and detectability modelling to identify which species can be surveyed with sufficient reliability to be able to characterise their relative abundances in different sites. And this has been done by Luke Einoder and his colleagues in NT Government's Flora and Fauna division, using a really great long-term data set in Kakadu National Park, where they've collected together all the data from the lowland savanna woodlands in the northern half of Kakadu and run these occupancies and detectability analyses. And what they've come up with is that out of the total of 146 vertebrate species, only 50 of them occur with sufficient occupancy and detectability to be reasonably able to be included in monitoring. And here they are, both so birds, reptiles and mammals, but also notably there are 2 threatened species there that are included in it. Now, this doesn't necessarily mean that other species cannot be included. What it does mean is that if you want to include them, then there'll have to be dedicated survey to increase their detectabilities to acceptable levels for them to be included. So we talked about animals being the poor cousins of plants in restoration ecology, and so I spend a lot of my lifetime talking about invertebrates being the poor cousins of fauna whenever we talk about fauna. So just as restoration often is seen as revegetation, fauna is more often than not seen as vertebrate animals. And so I need to have a big plug for invertebrates. They are 95% of all animal species after all, but also they are playing many of these key roles facilitating ecosystem restoration, very widely used as indicators, as we've just heard in the last couple of talks. But to do that, we need the target taxa. Now, we just heard that metabarcoding is a fruitful tool. At the moment, the reference libraries are insufficient, though, to provide meaningful information at the species level. And so until that happens, if we want species level information, it is a case of collecting them and sorting and identifying by hand. And because invertebrates are so diverse, it's obviously not feasible to do that to all groups. And so one needs to identify target taxa that are representative of invertebrates more generally. And we've done a fair bit of work on that in using the same sites in Kakadu, these long-term survey sites, but using data for invertebrates in them and asking the question, what subset of taxa can represent the spatial patterns of invertebrate diversity most effectively? If you wanted to measure, to take three taxa, which three? Taxa being family here. And so what we've done, this is a score sheet down here. So on the left, so we've got the candidate families down here. And on the left, we have, first of all, scores for sampling practicality, because it needs to be feasible to be able to incorporate any taxon. And so there's scores there. And then there's scores for biological representation. And we've got two different conceptual bases for representation. One is representing total invertebrate diversity. So which individual families whose species richness and composition most closely correlates with that of total invertebrates. That's one way of looking at representativeness. Another way is complementarity, where it explicitly looks to capture the full range of responses, full range of patterns of different invertebrates. And so one would start off with a taxon that best represents total invertebrates, but then others that represent the broader range of patterns. So very different. You can see there are different taxa in different cases, and you put them all together, get a total score. So if you're interested in total diversity, you'd look at ants, these flies and weevils. If you're looking at complementary diversity, you look at those somewhat different taxa. And my last comment about what taxa to include, and we've heard this yesterday in Chris Brady's talk, is that because Ranger is situated in the middle of Aboriginal-owned land, middle of a World Heritage area that's listed for its cultural values, we need to be considering culturally significant faunal species, if there are any, and this is an ongoing issue for discussion. The third thing we need to think about is what about? We've decided which animals to include, what do we measure? And there's two sort of fundamentally different sorts of measurements. One related to species level analysis. And I think we all have seen, and we saw this in the previous talk, the species composition is particularly informative here. And here's some examples of, oops, I'll go back, of the succession from some restoration where there's partial overlap in ordination space of sites undergoing restoration and reference sites, some strong overlap. And then this is the ideal situation, restoration success, where the restored sites are nested completely within the variation of your reference sites. It's also a powerful way of plotting trajectories. Good old word, trajectory. Here's some old data of mine from the original Ranger revegetation trials back in the 80s, looking at successional change in ant communities. And you can see that there's the reference sites up the top left corner of ordination space. And this is plotting a chronosequence of sites from just a few months old through to 13 years old. And you can see that there's a trajectory that's sort of it's sort of winding its way somewhere and maybe going towards reference sites, but little confidence that we're actually ever going to get there. So it's species level information, and then of course there's functional level information too, functional diversity. And I've just sort of noted here that we could have two sorts of functions. One's an effects function, in other words, the effects of that animal or group of animals on the ecosystem, and one of the functional groups that have been identified is something like the feeding gills of birds. But we can also have response functions, how animals might respond to various ecosystem properties, and the holodependent fauna is one that's been recognised as a particularly important one that needs to be managed. The fourth issue is similar. What do we mean by similar? We have to quantify it. And so Luke and his colleagues in NT Flora and Fauna have spent a bit of time putting together this acceptability matrix, where it's all about plotting the, plotting on the y-axis the proportion of sites, the relative similarity of a site to the reference condition, and then with the x-axis, the proportion of the rehab sites that achieve that. And the idea is that successful restoration is indicated by having either 60% of the rehab sites achieving 80% similarity, or 80% of the rehab sites achieving 60% similarity as a quantifiable measure of similarity. We also talked about feral animals at Ranger, no surprises here. And the fifth and final issue is the one of robust sampling methodology. And it is both a spatial scale at which one samples, and I think most of us think that a hectare scale is a reasonable one to look at. But then more importantly, the actual protocols and techniques that will produce statistically defensible results with the issue of detectability in mind. And I'll just show some data for some ant work that Steph Oberproler and myself did, just showing how sampling intensity can have a strong influence on the assessed restoration outcomes. And if you look at the left-hand side, so what we did is we sampled the Ranger rehabs on 3 separate occasions. And on the first set of bars is the results for just the first set of samples. The second one is we combined into 2. So the first 2 samples combined, and the third one is combining all 3 samples. And you can see just with one sample, it was sufficient to show that species richness at reference sites was a lot higher than at rehab sites. So there was no doubt about that. But it's interestingly that as you increase sampling intensity, the difference increases. And that's because it only took one survey to get pretty well all the species at the rehab site, but you keep on getting more of the species in the reference site with increasing sampling intensity. And that is because any diverse assemblage always consists of a relatively small number of common species and this enormous tail of rare species. And so if with low sampling intensity, that you basically just record the common species, you can provide a misleading indication of the true extent of the differences in diversity between reference and rehab sites. And so, yeah, Luke's team, well, Luke's left now, his team that remains at NT Government, spent a lot of time reworking old data to come up with the most efficient details of how you would sample to enhance, maximise detectability. And this is what they've come up with, which they're now rolling out as a new survey sort of design in Kakadu and other parts in this type of the world. So I'll just finish with that. Just a recap of the overall framework, starting off with appropriate reference conditions, identifying the species to be assessed, what about them to assess, what is the acceptable limit of level of similarity, and a robust methodology. And again, just to reiterate, it's really important to do this, both to give industry the clear goalposts and also the government regulators a clear guide of what needs to be achieved. Thank you.
I've got time for one quick question before afternoon tea, if anyone has one. Sean?
They're both Johns.
I was going to try to sneak in 2 quick ones. One on the periodicity of monitoring. What's an optimum for monitoring trajectories at Ranger? And the second one is, how can this process that you've derived come up with information relevant explicitly to management and its response? Yeah, so both those, I think the answer to this is the same to both, in the sense that this
is purely designed to assess the outcome at the end. It's not, so this project is not about assessing trajectories and then coming up with sort of feedback to managers about what needs to be done. And so these indicators and standards and data are all about the end assessment rather than how things go along the way. But I mean, I think the point you're raising is a really important one, is that given that these end results are likely to take 100 years or more, even under the best situations, and industry doesn't want to hang around for 100 years and government doesn't want to hang around 100 years before signing off, how could you possibly use a trajectory approach to indicate successful restoration? And that's, I think that's a question that is now with SSB and Ranger. And it's particularly challenged when we might have some trajectory information over the first 10 or 15 years, but we have no idea what happens after then. And we've got no empirical model data of achieving any trajectory, achieving the end reference point. But that's a big challenge for Ranger and SSB, as you are part of, John, well known.
I think
a workshop next week.
There is a workshop on that next week. I'm going to spend a fair bit of time talking about that. So I'd like to thank all the speakers. And I think it was a really enriching sort of look at the state of fauna and restoration and how they fit, how fauna fit into the restoration picture. And hopefully we can begin to think beyond the field of dreams concept and put fauna more front and center. And I hope everyone enjoyed it and enjoy your afternoon tea.